Okay, let's talk about the hypothalamus and the pituitary, the central nervous system aspects of the autonomic nervous system. Another vetty bit. This is a foal born the day before this picture was taken, but probably shouldn't have been born for another 4 or 5 days after this picture was taken. It's a premature foal. The foal's rectal temperature was 39.7 degrees. Fever, yeah, pyrexia. Why might it be pyrexic? Temperature autoregulation might not be as good as it could be because it's such a young foal. There's another reason why it might be pyrexic, and that's the reason why we were taking this foal's temperature at least 3 times a day. Infection, yeah. Once you start dealing with animals like this, infection is a big risk, and one of the earliest signs of infection is a spike in temperature. So it could be thermoregulation, it could be an infection. Let's look at thermoregulation, and let's see if we can explain enough about it to decide what we think was going on with that particular foal. You'll remember that we talked in the first lecture about homeostasis and about the keeping of the interior milieu, essentially the extracellular fluid of the body, keeping that at the same temperature, pH, ionic concentration, osmolarity, so that the cells that are bathed in that fluid are happy cells. That is controlled by an area in the brain which is the hypothalamus. And it's so called because it's under the thalamus, hypothalamus. The thalamus is one of those areas of the brain that has a bit of a strange non-functional name. Usually if you find an area of the brain that has a strange name, it's a big area because it was unfortunate enough to have been found before anybody knew what it did. The thalamus, bilaterally, are 2 big lumps that sit around in the middle of the brain. And so people found these 2 lumps and said, oh, thalamus, who knows why. And it was many years until it was discovered what they did. And then they found this lump under the thalamus, and they said, under the thalamus, hypothalamus. Isn't it easy? The thalamus is a big waypoint in the middle of the central nervous system. This is a slice through a rat's brain taken at this level. So right through the middle of the brain. If you look at the slice, these 2 big round things here, there and there, those are the left and right thalami, together the thalamus. Great big things, you couldn't miss them. And this area underneath is the hypothalamus. The third ventricle is a fluid-filled space, a CSF-filled space, which defines the midline of the brain in this region, and is essentially donut-shaped. It wraps around the thalamus in this plane. So you can see it here, and you can also see it, to a lesser extent, just up there, just above the thalamus. But the interthalamic adhesion, the 2 thalami, they don't communicate with each other, but they abut against each other, and they kind of squash the fluid out of the middle of the third ventricle to make it into a donut shape. So it's a donut that sits in this plane. Here we've got bilaterally tracts of white matter which contain nerve fibres. Those are the fornices, the left and right fornix. Here in green we've got the median eminence, which is the beginnings of the pituitary area. The hypothalamus is this area that's bounded under the thalamus and around the base of the brain. Okay, so what does the hypothalamus connect to? Well, here is a sensory neuron coming from the cardiovascular system, travelling up and entering the brainstem here. So, a nerve fibre that comes from the thorax enters the central nervous system in the brainstem. What nerve is it travelling in? What kind of fibre is it? It's travelling in the vagus, yeah. What kind of fibre is it? It's a visceral sensory or parasympathetic sensory fibre, depending on your preference for the notation. It's travelling up into the brainstem. You can go now. And from there, the information is being carried all around the brain. And this is a dog's brain, or half a dog's brain. You can see on this dog's brain the thalamus here. And so this area here under it must be the hypothalamus. And look, the information is going to the hypothalamus. It's also going to various areas in the medulla oblongata, where we have the respiratory and cardiovascular centres. And it's travelling to the amygdala and the cerebral cortex. So it's able to affect emotional state, arousal, and also raise itself to sensory awareness. Let's now look at the motor outflow, going back to the cardiovascular system. And this time, there are two motor pathways going to the heart. There's this one, which leaves the central nervous system in the brainstem and travels in the... Yeah, afferent nerve. Which nerve? Somebody said it, but not loudly enough for me to hear completely. Vagus, good. Travels in the vagus nerve and gets to the heart. This motor pathway travels down the spinal cord and gets out in the cranial thoracic region, a clue, and then travels down to the heart. It's the... Yep, go on. Sympathetic branch of the autonomic nervous system. Innervates the heart via... Does anybody know which nerves it travels in? What they're called? It's dead easy. You'll kick yourself when you don't shout it out when you hear. Okay, travels in the cardiac nerves. And we have a third motor output here, which I've represented as an arrow. This is the neurohumoral output, the endocrine output from the pituitary gland, which is linked into the hypothalamus. Okay, we know where it is. We know what it's connected to. What does it regulate? All those things, and a whole stack more, are regulated by the autonomic nervous system. Either via the medulla oblongata, in the case of relatively simple autoregulatory functions like blood pressure and respiration. Those things tend to have a single sensory input and a single variable that's changing. So if you imagine blood pressure, the sensory input comes from the baroreceptors. It either goes up or down, and the output is either an increase in cardiac output or a decrease in cardiac output to keep blood pressure the same. It's a straightforward negative feedback response. Respiration is the same, based around the stretch receptors and the central chemoreceptor trigger zone and carbon dioxide and so on. They're very straightforward, very basic control loops. The loops that are controlled by the hypothalamus are a little bit more involved than that, a little bit more subtle, and tend to have more than one sensory input and tend to have output at more than one level. So if we take the example of thirst, thirst comes from a dryness of the mouth, comes from osmotic receptors throughout the body, comes from the kidney, comes from all sorts of different places. The hypothalamus regulates and senses all that information and gives an output to various centres. We have this feeling of thirst. It alters our behaviour. We might stop off at the pub on the way home because we feel thirsty. Lots of different complex behaviours are playing a part in that regulation. And some of them, growth and emotion, are regulated in very complex ways throughout the life cycle. I want to look at temperature regulation for the rest of today's lecture. And the reason I want to do that is not because it's more important or less important than any of the other things that the autonomic nervous system regulates. I want to do it because in terms of the hypothalamus, it's something that we can consider as a unit without dragging in too much other physiology. If we start to look at thirst, then we're bringing in a lot of renal physiology and acid-base physiology and osmotic physiology. If we start to look at hunger, then there's a lot of gastrointestinal physiology that we need to drag into the picture. Many of these are very complicated. Temperature, we can at least put in a box and say, this is temperature autoregulation. So let's have a look at it and let's see how it works. In order to consider how animals autoregulate temperature, we need to know about the concept of core and peripheral temperature. The core is an area of the body that stays at, broadly speaking, the same temperature. It's very tightly autoregulated in terms of temperature, but it can get bigger or smaller. There are some parts of the body that are almost always part of the core. The brain is always core, the liver is almost always core, the cranial abdomen is usually core. And there are areas that can be core or not depending on how much temperature stress the animal is under. So the caudal abdomen, for example, is usually core under normal circumstances, but if you're foolish enough to go for a walk in a t-shirt on a cold day, your caudal abdomen will pretty soon no longer be core. Your core temperature area will shrink to the more important areas of the brain and the liver. The peripheral temperature is the rest of the body, and that is less tightly autoregulated. So, for example, the tip of a dog's ears can get a lot colder than their core temperature, or on a hot day can be at core temperature, depending on how the animal is using its surface to auto-regulate its temperature. And you will know, or at least if you travel on the bus, once the winter sets in, you will know that your toes and your fingers can get a long way below core temperature before a bus arrives. So, core is the central body compartment and has a stable temperature. Animals alter the amount of heat in the body by increasing or decreasing the size of this core as a heat store, effectively. And they allow the peripheral temperature to vary depending on the local circumstances. Why is regulation of core temperature important? Sorry? So you don't fry your organs, exactly right. Enzymes are the things that tend to fall apart first. And if your body temperature goes typically much above 42 degrees C, then enzymes will start to denature. And we're not talking about the pedestrian, put them in a test tube and look at them enzymes like gastrin and all those things, and trypsin, the ones that you study in A-level labs. We're talking about fairly major enzymes. Enzymes in Krebs cycle that generate energy for the body, enzymes in the muscles that allow muscular function, enzymes in the liver that allow you to detoxify things. Major enzymes will stop working if your core temperature rises above 42, and this is a bad thing. And we're also talking about structural integrity. If you get a nice fat steak and put it on a barbecue, what happens to the structure of the proteins that steak's made of? They denature. They go all sort of brown and crispy on the outside, and you think, that'll do nicely. But it's not good if you still need to use that muscle to get about the place. So the structural integrity disappears, and ultimately the animal dies. It's a bad thing. And so that's why animals thermoregulate. Within the hypothalamus, there is a hypothalamic thermostat, a group of specific cells within the hypothalamus that have a set point for response. And as the core temperature rises over this set point, they respond and they put in place cooling mechanisms. And those cooling mechanisms can be straightforward, or they can be fairly complicated. They can be as straightforward as peripheral vasodilatation to bring more blood to the surface of the body, to bring more heat to the surface of the body, to let it escape. Or they can be as complicated as seeking out a nice cool bar, preferably with a jacuzzi because it's too hot outside. Lots of different behavioural, physiological responses which bring that temperature down. But the hypothalamus centrally is all about bringing temperature down. That's what its job is. Okay, let's look at mechanisms of heat conservation. This is Jeremy, who several years ago now was a postgraduate student of mine. Jeremy wanted to look at a new way of measuring blood flow in tissues. And he was really excited about it, and we decided that the tissue he should use first was his hand. And we decided that a cunning way to reduce blood flow in Jeremy's hand was to plunge it into a bath of salted ice for, well, as long as he felt like, really, and I went to get a cup of tea, because that's what all good supervisors do. Jeremy could manage this for about 20 minutes, and this photo was taken about 2 minutes before his sense of humour failure, at which point he took his hand out and we started to measure perfusion. But what's going on in Jeremy's hand when he puts it into the ice? What's the hand doing to conserve heat? Vasoconstricting, yeah, reducing the blood supply to the area. Anything else? Piloerection? Yep. Not really that significant in Jeremy's hand, but if you're a furry dog, piloerection can make a big difference between how much heat you're losing in your periphery. What's important about both of those things is that they are local responses. Yeah? The hand is cold and the hand gets vasoconstriction, piloerection. The other hand's fine. It can be a completely different temperature, have a completely different blood flow and not have piloerection. So, mechanisms of heat loss are centralized. Mechanisms of heat conservation are localized to specific areas of the body. This is a place where I was a resident on a particularly nice day. I used to like that kind of weather. But it does give us a chance to talk about mechanisms of heat gain. How do we gain heat if we're cold? What do we do? Shiver, yeah? Anything else? Exercise, yeah. We shiver and we walk up and down. Generally, we're increasing metabolic rate. We're burning substrate, not because we want to do anything useful, but, well, it is useful, but you know what I mean, not anything mechanically useful, but because we want to turn it into energy to keep us warm. So we shiver, we increase our metabolic rate, we exercise, we do things, we get warm. And again, these are centralized behavioral things. You don't shiver with the bit of the body that's cold. You shiver generally in the areas of the trunk and where, if you're very young, the brown fat is. We don't exercise with the areas that are cold. In fact, if your hand's been in ice for 20 minutes, it can be quite painful to exercise with it. But you can kind of run up and down and jog a bit and generally keep warm. So again, they are centralized. Finally, mechanisms of heat loss. When it's too hot, how do we get rid of some of that heat? Yep. The classic physical mechanisms of conduction, convection, and evaporation. And for us and most mammals, evaporation means sweating. But for some animals that don't sweat, and the most famous example is the dog, it's panting. But it's moving the external environment, moving gas over a moist mucous membrane and sending perfusion to that mucous membrane so that we get evaporation, so that the heat disappears in the latent heat of vaporization. Cunning plan. Okay, let's talk about two clinical syndromes that involve thermoregulation, and let's talk about why they're important and talk about how they work and what they do. The first one is hypothermia. Okay, core temperature too low. Why is this a bad thing? Well, first of all, hypothermia is dangerous because temperature regulation is impaired below 34 degrees C, and if you let your core get to below 28 degrees C, you become unable to regulate your own temperature. The reason for that is that your body enzymic reactions are all thermoactive. They're all temperature regulated. The kinetics of enzymic reactions is temperature regulated. How many of you have done that study where you get some gastrin and a bit of meat in a test tube and you put it at 10 degrees C and 20 degrees C and 30 degrees C and you see how long it takes for the bit of meat to dissolve? People done that experiment or a similar experiment? Yeah? And the warmer you get, up to the point where the enzyme denatures because it's got too hot, the faster that reaction goes. And that reaction is the same for all of the enzymes of the body. So if your core temperature gets down to 30 degrees, then your metabolic rate is halved. And so your ability to increase your metabolic rate has a ceiling effect put on it much lower than it does when your core temperature is normal. The lower your core temperature gets, the less able you are to use metabolism to generate heat energy to get it back up again. That reduction in enzymic activity reduces the ATP available for the cells. Which group of cells are the biggest users of ATP in the body? The brain. So it's not a surprise that as your temperature drops, you get altered mentation, drowsiness, coma. So now you're not able to increase your metabolic rate to get your core temperature back up, and you're becoming less able to do those sensible behavioural things that will lead you to be in a better place. So you might think, oh, this is a nice walk in the afternoon. I don't need to go into that nice warm place. I'm just going to go for another mile around here. You start thinking badly, and slowly you get drowsy, you go into a coma. Because of the reduced metabolic rate, the shivering centre is depressed and you become unable to shiver. And in animals that are hypothermic, it's always, well, it's a good thing and a bad thing when they start to shiver. It's a good thing because everybody thinks, oh, I can shiver now, it's getting warmer, let's give ourselves a round of applause, that's great. But it's a bad thing because then they start to utilise oxygen at a much faster rate, and you better make sure you've got the airway and the oxygen delivery sorted out by the time they start shivering. Otherwise, they're going to run out of other things like oxygen, and life is going to get interesting in other ways. So the shivering centre is depressed, and hypothermia induces a stress response, a whoopee big stress response. And so we get all those catabolic, increasing metabolic rate enzymes dumped in the circulation to try and kickstart things as much as possible, and the animal gets into the classic stress response situation that we'll be talking about later on, or that John will be talking about later on. The opposite of hypothermia, in a sense, is pyrexia, increased core body temperature. And as we said right at the start, most commonly in adults, certainly, pyrexia is due to infection. And that's why we're so keen as clinicians to take animals' temperatures to see whether infection is or is not a likely etiology for the disease process that we're dealing with. Pyrexia is due to impaired function of the hypothalamic centres. The thermostat set point changes, it increases, so the animal's core temperature has to get higher before it starts to respond to cool it down. And once it does respond, the magnitude of that response is blunted. So the slope of that increasing response is reduced and the animal makes less effort to cool itself down. So we get both an altered set point and an altered response to increased temperature. Due to things that we call pyrogens. Isn't it easy? Pyrogens cause pyrexia. When you're dealing in somebody else's language, you can glue words together like that and there's nobody there anymore with Latin to tell us that it sounds horrible, but it works really nicely. So with pyrexia, we've got an impaired function of the thermostat, an altered response to increased temperature. And the pyrogens are things like tissue breakdown products, the kind of things that cells spit out when they get damaged. Arachidonic acid and its metabolites, histamine, potassium, protons, all those things are pyrogens. Bacterial toxins, especially endotoxins, the lipopolysaccharides, are really, really, really good pyrogens. If you want to see a really stoking pyrexia, give an animal some endotoxins and they will get really warm. Pressure, certainly within the central nervous system, can be a pyrogen. So if you see an animal that's got raised intracranial pressure, it might be pyrexic. And hand in hand with pressure goes ischemia. Central nervous system ischemia, or lack of oxygen supply, lack of perfusion, is a pyrogen. Now here's a basic physiological question for you. If you've got lots of different things having the same effect, is it likely that there's lots of different physiological mechanisms by which they have those effects, or is it more likely that there is a central clearinghouse and a single physiological mechanism that they all feed into? Any offers?
Single response.
Yeah, usually there's a single mechanism, because life is easier like that. So usually everything feeds into a single response. It is thought, but not really certain, that pyrogens all have an effect via a molecule which has been called endogenous pyrogen. And it is thought that endogenous pyrogen is something that's secreted by polymorphonuclear leukocytes. So when you get an inflammatory response, these polymorphs respond to the breakdown products of tissues, the endotoxins, all the other things going on, and secrete this endogenous pyrogen, which has an effect on the hypothalamic cells. Whatever the mechanism, it definitely involves eicosanoids in the hypothalamus. Eicosanoids are breakdown products of cell walls. And they are the things that non-steroidal anti-inflammatory drugs reduce the production of. So things like ibuprofen, Nurofen, aspirin, all those drugs reduce the production of eicosanoids and so reduce the inflammatory response, and so are antipyretics. They reduce pyrexia. There are two main classes of antipyretics. There are the non-steroidals, which reduce eicosanoid production by blocking the enzymes. And there are the steroids, which reduce the secretion of these putative endogenous pyrogens by the leukocytes. Both of those drugs have interesting side effect profiles. Generally speaking, the non-steroidals are used more than the steroids because the side effect profile of the steroids is really exciting, particularly in patients that have an infection. So, let's finish off by looking at the clinical role of antipyretics. Why might we want to use antipyretics clinically? If you've got a fever and you take yourself to bed, why do you take yourself to bed with aspirin or ibuprofen or whichever drug you choose? Lowers the fever. Yep, good. Anything else? You think to yourself, oh, I must lower my fever, so I'll take some aspirin. Is that the thought process that goes on? Comfort, yeah. You think, God, I feel rough as a scrote. I need aspirin. And you take aspirin and it makes you feel better, yeah? Okay. The important physiological thing is that the fever is reduced, but for the patient, the important thing is that the comfort is increased. So, antipyretics will also reduce morbidity. If you've got a patient with a body temperature that's bordering on dangerous, and you reduce that body temperature, then the morbidity or the badness, in inverted commas, of the disease is reduced. Does everybody know about morbidity and mortality? Yeah? Morbidity is a bit difficult because when an epidemiologist talks about morbidity, they talk about the number of animals that get a disease in a population, and when clinicians talk about morbidity, they have a much more woolly sort of badness term in their mind. You know, how morbid is this disease? How bad is it? How much am I going to really not like having this disease. So, antipyretics reduce morbidity. They normalise body function. So they put your body in a better space for its immune response to get on with the job of eliminating whatever the infective process is. And as we said, they increase the comfort of the patient. The non-steroidal analgesics have, over the last 10 or 15 years, found an increased place in perioperative management of patients for analgesia in the early post-operative period through to several days after surgery. But I think they're still underused in patients that come in that just generally feel ill and rough. And when we see 20 dogs in a day, all of whom have the same sort of infective illness, it's really easy to forget the fact that those animals are not feeling well and so they're likely to be a bit miserable. And giving them something as simple as an antipyretic can contribute to the management of their disease and also make them feel an awful lot better.